Hello everybody and welcome to the latest Asian Carp Canada webinar. My name is Rebecca Schroeder and I work on the Asian Carp program at the Invasive Species Center, and I will be your moderator today. So before we begin, there are a couple of items I wanted to touch on before I hand it over to our speakers. Firstly, there will be time for questions at the end of the webinar. If you have any questions at any point during the webinar, you can type it into the question box and I will read it out loud to our speakers at the end and they can answer them. Also would like to note that we do have Dave Marson available from Fisheries and Oceans Canada. If there are any Canadian-focused questions that you'd like to ask throughout the webinar, he will be available at the end. Secondly, if you're having any issues during the webinar at any time, you can write them in the chat box or the question box, and I will try and resolve it for you, or alternatively, you can send me an email. When you registered, you probably got my information. And lastly, there will be a brief survey at the end of the webinar, so if you could take a few minutes to fill that out, it would be greatly appreciated. It just helps us make sure future webinars are what everyone's looking for and what everyone wants. So today's webinar is titled Grass Carp in Lake Erie, History, Status, and Habitat. I am pleased to introduce our speakers, Dr. Patrick Kachowski and Nicole King. Dr. Patrick Kachowski is a research fishery biologist for the U.S. Geological Survey stationed in Sandusky, Ohio. His work on grass carp began in 2012 with a research paper cautioning that the Sandusky and Maumee rivers were suitable for spawning of grass carp. That was followed by a 2013 paper reporting recruitment of grass carp from the Sandusky River. Subsequent research identified the Maumee River as a spawning river and an assessment of past recruitment in the Maumee and Sandusky rivers. Patrick is the USGS lead researcher for grass carp on the U.S. side of the lake. Nicole R. King is a research associate at University of Toledo Lake Erie Center in Oregon, Ohio, and has been working on the grass carp project since 2015. Current research focuses on the early life of grass carp, including identifying spawning locations and spawning cues in Lake Erie tributaries. She is also working on mapping aquatic vegetation throughout the U.S. side of the western basin of Lake Erie to identify preferred grass carp habitat. And with that, I will pass it over to our speakers.
Yeah, thanks for having us. This is, it's exciting to be part of this webinar series. I think I'm going to do most of the talking, and Patrick will chime in whenever he feels the need.
Hear, hear.
We're not advancing. Sorry, we're having some trouble advancing. There we go. Okay, so before we start, I wanted to thank everyone that has worked on this project, but there are way too many agencies and way too many people to thank individually. But I did want to mention that everything I'm presenting today is really a multi-agency effort. So all of these agencies contribute little bits and pieces to the bigger picture here. And then a real thank you to all of the technicians that have worked with us over the years. They really do a lot of the hard work, and then they don't get much credit. So a big thank you to all of our techs. Okay, so this is something that I've been starting most of my recent presentations with, and that is that the term Asian carp is really misused. So Asian carp is not a species. It's actually this umbrella term that refers to this group of species. But when you hear the term Asian carp, what usually comes to mind is silver carp, and those are those videos you see of those jumping fish that hit fishermen and end up in boats. But that is actually the only species in this group that exhibits that behavior. Every once in a while, you'll hear Asian carp as a group for the silver carp and the bighead carp, and I think that's because they look similar and they eat similar things. So these fish are both planktivores, and these are the two that everyone's really concerned about getting into Lake Erie because they have the potential to kind of wipe out the bottom of the food chain. But all of these fish are pretty different. So grass carp are herbivores, they consume aquatic vegetation, and black carp are molluscivores. So they're eating things like clams and snails. And so really this term kind of gives me nightmares. It's giving me gray hairs, honestly. And that is because it's really, really misused, especially in the media. So all of these articles here have Asian carp in the title, but this first article here is actually about silver carp, but it's showing a picture of a black carp. This article is actually about silver and bighead carp, and they have a picture of common carp, which does not even fall under the Asian carp category. And here you go. Here's an article about silver carp with a picture of a bighead carp. This article is actually about eDNA. It doesn't even list the species, so I'm not even sure what species this is talking about, but there's a picture of a silver carp. And then this article is about grass carp with the ever seen photo of a silver carp jumping. So in the future, I would love it if everyone could just use the species name of whatever fish they're talking about. Okay, so although this term Asian carp, and you'll notice I tacked an S on the end of there, so maybe it clues people in that I'm talking about more than one species, but although you see a lot about Asian carps in the news recently, they're not necessarily new to the US. So these species were brought into the US in the 60s and 70s for use in aquaculture ponds, and they eventually escaped into the Mississippi River Basin during flooding events. In the Great Lakes, as of right now, there have been no observations of black carp. As for silver carp, there has been some eDNA collected in Lake Michigan and in Lake Erie, but so far no captures. There have been, interestingly, 3 bighead carp captured in Lake Erie, one in 1995 and 2 in 2000, but no captures since then and no eDNA. And then grass carp are, as you can see from that map there, these are pretty widespread throughout the Great Lakes. They've been captured in every Great Lake except Lake Superior. So from now on, I'm just going to talk about Lake Erie grass carp for the rest of this presentation here because these are the ones that are really widespread. And I'm probably preaching to the choir a little bit here, but grass carp are an invasive species of fish, and they can consume large amounts of aquatic vegetation. In some instances, up to 100% of their body weight per day. And because of that feeding, they can do some real damage to wetland ecosystems. And whenever you have damage to these ecosystems, you of course can have impacts on native fish species and vertebrates and even waterfowl. A lot of these waterfowl species rely on submerged aquatic vegetation as food when they are migrating. And then there's some evidence that suggests that grass carp may promote algal blooms, which is certainly something that Lake Erie does not need more of. So this is the current world record grass carp. This is a gravid female that weighed in at I think 92 or 93 pounds. So you can imagine that this fish throughout its lifetime has consumed quite a bit of vegetation. Okay, so again, you see grass carp in the news a lot recently, but they're not necessarily new to Lake Erie. They were brought over to the US in the 60s. And then it actually took 20 years for the triploid process to be created. So in some states, you can purchase sterile triploid grass carp. These cannot reproduce. They're sometimes sold under the name white amur. And then the first grass carp was actually collected in Lake Erie in 1985. So at first, everyone thought, we have grass carp in Lake Erie, but it's not going to be a big deal. They either cannot successfully reproduce in Great Lakes tributaries, or these are sterile triploid individuals. The first case that, this might not be true, we might have a problem here, came in 2012 with the collection of 6 juvenile grass carp from the Sandusky River. The otolith microchemistry from those fish came back indicating that these fish were probably born and spent most of their time in the Sandusky River. And then we had our first direct evidence of grass carp spawning in any of the Great Lakes in 2015 in the Sandusky River. Holly Emke, a graduate student at UT, actually collected 8 fertilized eggs. So since then, we've continued sampling of Lake Erie rivers. And this is a kind of like a heat map. This was downloaded directly from the USGS Non-Indigenous Aquatic Species Database. And you can see that each of those dots represent grass carp captures. The bigger the dot, the more captures. And you can see that they're pretty widespread throughout Lake Erie, but a lot of those captures are concentrated in the western basin of Lake Erie. So over the past few years, we've really focused on tributaries in the western basin, mostly the Sandusky, the Maumee, and we've also done some poking around in the Portage and the Huron rivers in Ohio. And then as any of you are out there doing work or fishing in Lake Erie, if you happen to come across a grass carp, please report your sightings either directly to the USGS NAS database or to your local Natural Resources Department. Okay, so after Holly, our grad student, collected these first eggs, we had quite a few questions. And so I'm going to kind of take you through some of those questions and how we answered them. So our first question was, when do grass carp spawn? And after Holly's initial 8 eggs, we probably need a couple more eggs to really draw any conclusions. And so this is how we sample right now. That's just your standard 16-foot john. There is a custom arm on the front there that we use to tow bongo nets at specific depths. And what we found so far is that spawning can occur multiple times a year. This is just from the Sandusky, but you can see that spawning occurs throughout May, June, and July. And this picture up here is actually the cod end of one of our bongo nets. And so you can see there's some grass carp eggs poking up through the river debris up here. But this sample actually probably has several hundred grass carp eggs in it. They're just a little bit hard to see with this sediment. And then here's a grass carp egg on my hand. They're actually pretty big. So in the field, you can usually see these pretty easily. They're a lot bigger than a lot of our native species eggs. Okay, and what we found throughout the last couple years is that these fish really tend to spawn during high flow events. So this is a discharge from the Sandusky River in 2015. You have your discharge here and the dates down here. And the dots are dates that we sampled and did not collect eggs. The stars are dates that we did collect eggs. So what you can see is each time that we collected eggs, that spawning was associated with a high flow event. And then this dotted line down here becomes important a little bit later in the presentation. This is the minimum flow required to keep eggs suspended in the Sandusky River. So these eggs are semi-buoyant, so they don't float. They sink really, really slowly. And so they need a certain amount of flow and also turbulence to keep these eggs in suspension. So once these eggs sink, and if they get covered with any sediment, the probability of them hatching really decreases. It's not impossible, but it really decreases. So ideally, these eggs would stay suspended in the river to hatch successfully. So I came onto this project in the fall of 2015, right after Holly collected these eggs. And I was running my own crew in 2016, and I was all excited to go out there and collect eggs and get more data. And even though I was out on the river multiple days a week, I did not collect a single grass carp egg, which is good, I guess. But when that's what you're looking for, it can get a little bit boring. But when we look at the hydrograph, it actually makes sense that we didn't collect any eggs. So you can see 2016, it was a really low water year. We really did not have any peaks in the hydrograph. Only one time during our field season, it exceeded that minimum threshold. We did sample that day, but did not collect any eggs. There was just, it looks like the fish just did not spawn in 2016 because there were no high flow events. 2017, however, was a bit of a different story. This is the year that I call a go big or go home year. We had 2 peaks in the hydrograph, collected eggs during both of those, and actually during this mid-July event, we ended up collecting several thousand eggs just in one day. 2018 is a bit of the same story, 2 peaks in the hydrograph. We collected eggs during both of those peaks. And that brings us to 2019, which honestly was a little bit of a disaster. The whole spring and into early summer was a high flow event. So we never had like a nice peak like we see in other years, it was just a constant peak in the hydrograph. But we did collect eggs during several different events, and then also a later event towards the end of July. And if any of you were out on the rivers or on Lake Erie, you might remember we had a really, really high water year. All of our launches were underwater. This is my poor technician, Michaela, that had to wear chest waders every time she backed the truck in just to try to get the boat far enough back in the water. So good job, Michaela. Okay, so the next question we had was, are these eggs viable? Do we even have anything to worry about? Unfortunately, the answer is yes. These eggs are fertilized and are developing. So once we collect eggs in the field, they're preserved. We bring them back to the lab, put them under the microscope, and you can actually stage these eggs according to this publication from Yi and others. And then the big question next was do these eggs hatch in the river? So if you remember, I talked a little bit about the need for these eggs to have flow and turbulence, and that these eggs want to hatch in the river to be successful. So this is kind of a snapshot of a spawning event from July 12th of 2017. You can see that we collected a range of eggs. The earliest stage egg was a stage 3 egg collected right about downtown Fremont, and as you move downstream, these eggs are developing. And right before you reach the river mouth, we ended up collecting a stage 25 egg. And for reference, these eggs hatch at stage 30. So this egg was pretty close to hatching. It may have hatched in the river. It may have gotten blown out. But one of the things that we look at is that eggs are going to behave differently under different circumstances. So the development of these eggs is completely based on temperature. So warmer temperatures, these eggs are going to be developing faster. But the location of an egg in the river is based on flow. So this is that same 25 or stage 25 egg that I was talking about. This egg was collected when it was about 70 degrees Fahrenheit and with a flow of about 9,000 CFS. So the river was ripping this day. But on a different day, this was actually in, I think, July of 2018, we ended up collecting a stage 26 egg much farther upstream. This was several river kilometers upstream than our stage 25 egg. And this was under a similar temperature, but you can see that the discharge on this date was a lot less. So this egg kind of had a lazy river ride as far as development goes, and this stage 25 egg was pretty much getting blown down the river. So one of the ways that we can tell whether eggs would or would not hatch in the river is by using some modeling. And this is what we've been using. This is called FluEgg. And all this requires is basically a hydraulic map kind of uploaded into FluEgg. So you need to know what river you're on, and then you can select silver carp, big head carp, grass carp, or black carp are here also, just not shown here. And you can tell FluEgg where the spawning grounds are. You can tell it the temperature and the flow, and FluEgg will tell you whether or not those eggs would hatch in the river and where they would hatch. So this has been pretty useful. There is another model coming out. This was done by Thage, who is a grad student at University of Toronto. And he has a really, really nice animation, but I wasn't sure if it was going to work here, so I just took some screenshots for you. But the FluEgg model is a one-dimensional model, so it really just takes into account discharge. This model that Thage is working on takes into account some 3D hydrodynamics of the river. So this model can show low velocity areas and can actually show that sometimes eggs get stuck in these low velocity areas or get stuck in eddies, and then will eventually get resuspended into the river. And what that means is that some eggs end up spending more time in the river than others. So even if you have a flow event that might blow a bunch of eggs out down the river, and most of those eggs won't hatch, since this egg has spent more time in the river developing, the probability of it hatching in the river really increases compared to the rest of its cohorts. Okay, so our next question was, where are the spawning grounds? The way that flu egg works is you need to know the spawning grounds to estimate whether or not eggs would hatch in the river. But Holly, when she first got those 8 eggs, did not know where the spawning grounds were. So she actually used flu eggs a little bit differently, and it's not that she ran it backwards, but she had to kind of guess where the spawning grounds were and then run flu egg until the results from that matched up with what eggs she had collected. So she estimated that the spawning grounds for the Sandusky were right about downtown Fremont. In this photo here, this purple area indicates the 95% confidence interval, and this red circle here is the centroid. So then in 2018, something really, really cool happened. This sort of thing does not happen in field biology all the time, but the stars were aligned, or Mercury was in retrograde, or something happened, and the Ohio Department of Natural Resources hosted their planned action grass carp removal effort. I refer to it as the grass carp rodeo, because all of these different agencies from the US and Canada kind of descended on the Sandusky River with the objective of removing as many grass carp as possible. So this was scheduled weeks in advance, and it just happened to, when the date came, was a high flow event. And we all know now what happens during high flow events. So there were a lot of fish in the river, and over a 3-day period, these crews ended up removing 28 fish. That's what all of these little red dots are here. And the majority of those fish were captured right within the estimated spawning grounds. So that was really cool. And then to make this even cooler, as all of these crews were out collecting these adult fish, I was out there in my little 16-foot John boat, trying my best to stay out of the way, but also looking for grass carp eggs. And my crew and I ended up collecting a newly spawned grass carp egg just upstream of Highway 20, which is just downstream of where all of those adult fish were captured. So I think we have a pretty good idea of where the spawning grounds are in the Sandusky River. But that might change. So in the fall of 2018, an impassable barrier just upstream of our spawning grounds was removed. So the Ballville Dam was removed. This was really great in terms of news for the Sandusky River. This opened up a lot of habitat for walleye spawning. Unfortunately, it could also open up habitat for grass carp spawning. We don't know if that will happen or not, but it is something that we're keeping an eye on. We are also waiting for new inputs to flu eggs, since there may have been some changes in river hydrology. Okay, so before we move on to our next question, we did get a little bit more bad news in 2017. So we also documented spawning in the Maumee River. We really don't have as much information from the Maumee as we do the Sandusky, but it looks like the same patterns hold true. There's spawning occurring May, June, and July, and also associated with high flow events. So we really haven't spent as much time out on the Maumee. That will be changing this next year. And then even a little bit more bad news, we did end up collecting 6 grass carp larvae from the Maumee River in 2018. These larvae were collected pretty close to the mouth of the Maumee River, but this is proof that eggs can hatch, at least into larvae, in the Maumee River. There's no telling on whether these fish would have grown into adults or not. We just don't know that, but this does indicate that there is a possibility for eggs to hatch into larvae in the river. Okay, so what's being done about this? There have been several different publications coming out, a lot of risk assessments, but Ohio DNR just came out with this grass carp response strategy just this past year. This is available for download online for free if you're interested in reading it. And this publication outlined a couple of objectives. And the first was securing the aquaculture supply chain and bait, so making sure that no new fertile fish are entering into our lakes. Furthering research about life history, monitoring, and reducing populations. Another objective was removal efforts, which I'm going to get into more in a second. And then the evaluation of potential barriers, with the thought that if we can put up a seasonal weir in the Sandusky, you can prevent fish from getting up into the river and spawning in the first place. Okay, so this is relatively new. There are several what we're calling strike teams out on Lake Erie right now. Just this past year, University of Toledo and Ohio DNR created kind of a mixed crew. Michigan DNR has a crew, as well as Fish and Wildlife, and I know there are some crews up in Canada doing surveillance and trying to capture adults. So the kind of main objective of these strike teams is to be out several days a week trying to remove as many fish as possible. And these crews are using shock boats, nets, they're doing some live tracking on receivers. And then also once these fish are removed, they're processed. So we're getting a lot of really valuable information from these fish. There are several different structures taken for aging and otolith microchemistry. Guts are being taken out to look at diet. And these crews have been pretty successful this past year. They've managed to remove over 150 fish just this year. Whoops. Okay, and this is just to highlight one of the really interesting things that we found out from removing these fish. So this was done by Sabrina, who is a grad student at UT, and she took the ages of these fish and back calculated birth year. So you can see that there were a lot of fish that came out of the 2011 birth year. And if we look at the hydrograph from 2011 from the Sandusky, you can see that there were a lot of peaks in the hydrograph, a lot of high flow events. So these fish really had plenty of opportunity to reproduce that year. And that red line that you see there is that minimum flow threshold required. Okay, so there's also a lot of other research efforts going on. There's a lot of early detection and surveillance research, a lot of work going on about identifying spawning grounds, both in the Maumee and in some other rivers, whether or not spawning is happening in other rivers. A lot of modeling going on. And right now we don't have a really good population estimate. So there's a lot of work going on with that. I know both at UT and at Michigan State, University of Michigan.
On what aspect?
Of a population estimate.
Yeah, the Quantitative Fisheries Center at Michigan State University is working with telemetry data and capture data to try to come up with a way to estimate the population size.
Yeah. And yeah, a lot of research going on capture probability and gear efficiency. I know USGS is working on some baits and attractants with the thought of if you can attract a bunch of grass carp to one place, that's a lot better removal wise than chasing individuals around Lake Erie. And then again, your seasonal barriers. Okay, so this is not all doom and gloom. I don't want anyone to get depressed here, but just because we're collecting eggs and larvae does not necessarily that means all of those individuals would have recruited to the population. We really don't know how often these fish are successfully reproducing and recruiting. We don't know what the population is right now. What we do know is that these fish can do some real damage to aquatic vegetation and our wetland systems. And there's a real need to continue this type of work and eventually to try to remove all of these fish. The one thing that's kind of a silver lining, I guess, is that grass carp have really similar spawning requirements to other carp species. So whatever we learn about grass carp spawning gets us ahead of the game, say if silver carp end up making it to Lake Erie. Okay, so that was kind of a brief overview of what we know about grass carp spawning in Lake Erie, but I'm kind of gonna change gears here and highlight some of the habitat work that we have been doing. We know that these fish can alter wetland ecosystems, and we really want to know what's out there and where this vegetation is, so we can potentially track any changes from grass carp herbivory. So I've been working on this with several people, but especially Jenny and Travis from USGS UMass. Okay, so the way that we're doing this is using a 3-tier assessment, and I'll get into each of these methods shortly, but these 3 methods that we've chosen are OBIA, or object-based image analysis using aerial imagery, hydroacoustics, where we're out there running a sonar unit, and then also good old-fashioned rake surveys. And so we've chosen these 3 methods because each has kind of their individual pros and cons, but by putting these 3 methods together, we think it gives us the best picture of what's out there. So OBIA is really great for large-scale mapping. Lake Erie is really big, so we need to be able to map large areas. And then the OBIA provides a really good starting point for likely submerged aquatic vegetation location. So I'm not just throwing darts at the map and going out and sampling in one area or another. It's also pretty fast. We can get some pretty fast results from that method. And then, unfortunately, OBIA tends to use outdated imagery. So if I need to be out there on the water sampling vegetation in July of 2020, I'm probably not going to have really current maps from July of 2020. Then moving on to Sonar, this really provides accurate detection of SAV, but there's no way I'm going to be able to scan the entirety of Lake Erie. It's pretty time consuming, and neither OBIA or Sonar offers any community data. The rake method, however, does provide community level data, but it is extremely time consuming and really only gives you point data. So by combining these 3 methods, we get a pretty good picture of what's out there. So as for OBIA, I'm kind of just going to walk you through the steps that this takes. And the first step is probably the most labor intensive, and that is scanning through all available aerial imagery and trying to find good photos. So after those are collected and bathymetric contours are downloaded, all of this is brought into a program called eCognition. And this program uses variations in light to pick out differences in your photos. And then the user can create a rule set that's used to classify submerged aquatic vegetation. And then eventually those results can be exported. My co-author Jenny, uses ArcGIS where she adds density modifiers. I import these results into Google Earth, and that's because I can take Google Earth with me on my phone when I'm in the field and kind of see these things live. So what you see here is a zoom in of the western basin of Lake Erie, and all of these areas that are marked in red are areas where that program has indicated aquatic vegetation exists. And then this is kind of interactive, so you can click on these polygons and it'll give you more information about what's there. So EM is emergent vegetation, SV is submerged vegetation, and then the letters at the end are your density modifiers. So A, B, C, and D all correspond to a certain percentage of density. And then this is a zoom in of an area to just give you an idea of what it looks like. This is the north side of Kelly's Island. And here you can see that there's several different polygons. Each has that label with the density modifier. So then I can actually bring in and overlay my hydroacoustic data. And so you can see that these green transects here are the transects from my hydroacoustic unit. We're using a Biosonics MX downscan echo sounder. And this is also interactive, so you can click on any of these bins and it will bring up a specific density that shows plant coverage. So the more dark the green, the more dense your vegetation is. And so then we can kind of compare densities from the OBIA to the hydroacoustic transects. The last thing that we're doing is the vegetation surveys, and this is by far the most time consuming. There's a field component and a lab component. So usually my poor technicians are up at the front of the boat chucking rakes all day. We bring those samples back to the lab, sort everything to species, and then everything goes in the drying oven so we can get biomass. And then one of the things that we're doing with this data is trying to find grass carp preferred habitat. So what we've done is combed through the literature and tried to figure out which species of plants that grass carp prefer to consume. So you can see here that they prefer a lot of different species. Maybe they don't prefer coontail so much or milfoil, but what we end up doing with this is creating a heat map. So the lighter green colors indicate areas of more preferred food sources, and the larger the circle, the more biomass is there. So if I were a grass carp, I would probably choose to hang out by the Bass Islands here, South Bass, Middle Bass, North Bass. You can see that there's a lot of really preferred food sources there, and also there's quite a bit of biomass. If you compare that to Kelly's Island, there's a lot of biomass there, but it's not a really preferred food resource. This was a lot of coontail there. Okay, and then one of the other things we wanted to do with our data was compare how well each method can identify vegetation. And so the way that we are looking at this now, this is all preliminary results, but we're looking at agreement between species. And so that's based on how well these methods match up with presence and absence of SAV. So rake sampling and hydroacoustics had the best agreement at about 93%. Rake sampling and OBIA had an agreement of 69%, and hydroacoustics and OBIA had actually pretty low agreement at only 54%. But that agreement increased to 72% when SAV density was over 50%. So if the hydroacoustics indicated 50% cover or more, OBIA was much better at detecting the presence of that SAV. So what this means is that hydroacoustics is really good at detecting the presence and absence of SAV, where OBIA has a little bit of difficulty detecting SAV if it's sparse or patchy. And then the rake sometimes misses SAV when it's sparse or sporadic. So if your vegetation is really patchy, you might not throw the rake directly over that patch. Okay, and this is really why we're doing this. This is just baseline data right now, so we can track any changes in distribution, density, or community composition potentially caused by grass carp herbivory. There are other invasive herbivores out there, like the red swamp crayfish, and then also SAV communities can change from year to year. So it'll be a little bit difficult to say that it's due to exactly, grass carp, but we do want to be able to track any changes that are out there. We are also working, comparing data and methods with others. We are not the only crews out there doing SAV surveys. So we will be comparing our data and methods with the Nature Conservancy and Cleveland Metro Parks. We worked with both of those agencies this summer, and when we get around to analyzing everything, we'll get together and I guess see how well we did. So with that, I am happy to take any questions.
Well, thank you very much, Nicole, for that great presentation, and Patrick for chiming in. So yeah, we do have some time for questions, so I'll get us started. Why haven't we run flu egg for the Maumee River or on the Sandusky River for 2019 spawning?
Yeah, this is Patrick. I'll take that question. The hydraulic model for the Sandusky River needs to be extended before we can do any work on eggs from 2018 or 2019. The existing hydraulic model ended at Ballville Dam. New data had to be collected farther upstream and downstream of that dam to update the hydraulic model to determine if there are any changes in hydrology. That work should be completed in middle of 2020, at which time we'll be able to run the model and determine if there has been a change in the spawning area or if any new spawning areas have occurred. The model for the Maumee River existed in certain parts of the river, but not for as long of a reach as we thought necessary. So data were collected for updating and improving that model. We expect that work to be done later this month or next month, after which we will be able to run the flu egg model for the Maumee River and estimate spawning areas from 2017, 18, and 19.
Okay. The next question is, what is the status of Theorem or other targeted toxins?
None of us are working with targeted toxins, so we can't directly answer that question. That'll be better answered by persons at the USGS labs in Upper Midwest Environmental Science Center and Columbia Environmental Research Center, who are working on attractants and baits. As far as direct toxins, I'm not aware of anyone in our organizations who are working with toxins specifically for grass carp.
Okay, next question. In the year that had eggs released in both June and July, were the eggs in July fertilized?
Yes, all of the eggs that we collected have been fertilized.
Okay, next question. Are grass carp spawning in other rivers besides the Sandusky and Maumee River?
We don't know that right now. So the Sandusky and the Maumee are the only rivers that we, in the Great Lakes, that we have collected eggs in. I mentioned that we also did some sampling in the Huron and Portage River, but we did not collect any eggs during those sampling events.
Okay, next question is for Dave from DFO, if you're still on the line. We're wondering if you can give just like a high-level overview of the status in the Canadian waters.
Yeah, I'm still here. You can hear me?
Yep.
Okay, yeah, we actually have had a quiet year this past sampling season, so no fish were captured, no grass carp were captured during our early detection surveillance, and there were no captures through commercial fisheries either in the Canadian waters of the Great Lakes. So it was a quiet year. We did have success capturing fish when we went down to the Sandusky to help out with the efforts there. So DFO did capture fish, but not in Canadian waters.
Okay, thank you.
Yeah, Rebecca, this is Pat. I just add a commentary.
Yep.
Yeah, so binational support for research and management in the Great Lakes is a cornerstone of success of the past 50 years. Just want to extend our thanks to Fisheries and Oceans Canada and the Ministry of Natural Resources and Forestry for working with us in the Ohio waters of Lake Erie on management. been a great collaboration.
Awesome. The next question is, once all of the adult grass carp have been processed, what happens to them? Are they provided to food banks for human consumption, or what is done with the removed biomass?
So once the fish are processed, they're just disposed of. Unfortunately, these crews are out there capturing fish all day. The fish do go on ice, but they are not fit for human consumption because we'd have to follow certain guidelines for that about safe handling.
Okay. The next question is, what is the greatest policy gap at the moment that could lead to recruitment?
I'm sorry, could you repeat that? Did you say what is the policy gap?
Yeah, what is the greatest policy gap at the moment that could lead to recruitment?
I don't think I can address policy gaps related to recruitment.
Fair enough. The next question is, have you analyzed the spread or the genetics to determine how the fish became so widespread in the lower lake?
There are no analyses underway that I'm aware of to address that. We do have some genetic data from eggs and adults. We had used genetic data a few years ago to try to get a ballpark estimate of the number of fish that spawned in the Sandusky River, but I don't know of any efforts that address the question that you posed.
Okay, we have one more question as of right now, so if anyone has any more, ask them now while this one's being answered. So the next question is eradication of grass carp in Ohio's waters a realistic and achievable goal moving forward?
Yeah, that would be a question for Ohio DNR to answer. I don't believe a DNR representative has voice access right now, but that's something that's not within my purview to answer.
Okay, we have one more. Canada determined that the river in Port Arthur could support grass carp. Are other rivers in Superior going to be assessed for risk in the US or Canada? That might be like a two-way question.
Yeah, so within Lake Superior, I know that the Kolar et al. 2007 book addressed rivers throughout the Great Lakes. It has not been targeted in the US because there has not been a single grass carp reported from Lake Superior, but I do imagine future efforts will look at the potential suitability of US tributaries to Lake Superior.
And then with the Canadian side, we did have a couple of sites in Lake Superior that we were sampling, but the threat is clearly higher in the western basin of Lake Erie. So we've adjusted our surveillance to target areas closer to the western basin to put more effort into those areas closer to populations of grass carp.
Okay, that's all the questions we have. So if there are any more, it looks like Nicole and Patrick's contact information is there. You can also send me an email and I can see about getting any of your other questions answered for you. Oops, sorry. And so with that, I'd like to thank Patrick and Nicole for speaking to us today and everyone for tuning in, and also thank you to Dave for joining us to answer a couple Canadian-focused questions. The webinar was recorded and will be posted on our website most likely this afternoon. Our website is www.asiancarp.ca. And again, please, if you have time, it would be really helpful if you could take the short survey that we have that follows this webinar. It really helps inform future webinars. So thank you again for listening, and thank you again to our speakers, and stay tuned for some future webinars.
Thank you, Rebecca.
Thanks for having us.